Thank you very much. Yes, so it's a great pleasure for me to give a talk to you here at the International Society for Biofabrication Seminar Series. So first of all, I would really like to thank the organizers for the invitation and especially providing this amazing platform when we can come together to do to introduce our research virtually. So today I would like to talk to you about some biofabrication technologies developed in my lab. They are from 3D fiber printing to MOrPFing life-size organoid tubes. So here in this slide shows the roadmap and also the vision on how biofabrication and bioprinting is moving forward as a field. So it proposed to start from 3D printing models for biomedical modeling and surgical planning, to the second stage, creating permanent implant, and to the third stage, making biocompatible, biodegradable tissue engineering scaffolds, to the fourth stage, developing in vitro models using cells and building blocks, and finally to the fifth stage, generating mini organs with biological functions. In all of these biofabrication examples, if we would like to mimic one or more functions of the biological system in real life, for example, in the case of surgical modeling, a good model could integrate anatomy and mechanical property of the organ of interest, but the biological components may not be necessary at all. On the other hand, for in vitro models, biological and physiological functions are very important, and actually they are essential. So the above concepts really guide the research activities in my lab. As an overview, my group has developed several technologies to process and structure functional materials and biological matters across length scales in three dimensions. And these are split generally into four main areas. So in the vertical axis, we've developed unique capability to manipulate and print small fiber structures in 3 dimensions. Then crossing to the macroscopic scale, we design new methods to print soft material and hydrogel-based materials. Then moving to the horizontal axis, we integrate tissues with devices to create microvessels to study their interaction with cancer cells, and also build organoid on-a-chip devices for biological investigations. Now, working with biological and living systems, self-assembly and also tissue morphogenesis become naturally part of the biofabrication workflow, and this is what we really focus on as well. Now, for the outline of my talk today, I will first introduce the capability we developed for 3D printing of high-resolution fibers. And second, I will demonstrate the physical insight that we developed to investigate the interaction between cells and fibers. And here, in this case, I call the cells the free spirit. So third, I will show you a new bioassembly approach called MOrPF, MOrPFing, MOrPF, okay? So this MOrPF process can upsize and also shape organoid tubes. And I will discuss my thoughts on when we should engineer a living system and when we should actually just leave the living system, the biological systems, to develop by themselves. And finally, I will demonstrate how we can combine 3D printing and machine learning to model clinical readouts of cochlear implant patients, and therefore taking modeling to gaining new insights into the human body. So I will start with the topic of 3D printing of high-resolution fibers. As a general overview of fiber patterning, many advanced technologies have been realized for thermoplastic fibers. And here, this image is showing some really beautiful fiber structures made with melt electrospinning and also electrohydrodynamic writing. Different methods can be used to stabilize a charge jet, building other fiber architectures. And therefore, the resulting architecture determines the biological response or the actuation response. And here, the focus for thermoplastic polymers are for their architecture. Now, in my lab, our focus is placed on enriching the functional materials processable beyond thermoplastic materials. So based on solution fiber spinning, we could start look at for example, in situ chemical reaction and also self-assembly of materials in the fiber itself. So the composition of the fibers could be synthetic, naturally derived, biologically derived, or they could be composites. And with the variety of materials available, the resultant function of these fibers could be used for reinforcement, therefore mechanical. They could be bioactive, they could be piezoelectric, conducting, or fluorescent. So now, taking back a little bit, it is clear that the processability of a material will be dependent on the material composition. An interesting analogy here is comparing the processability between molten cheddar and also molten processed cheese. The molten processed cheese has much more regular and predictable flow property than the molten cheddar. And therefore, as you can see on these dishes, the molten processed cheese can really create very beautiful, intricate, and well-organized structure on the dish, while the molten cheddar is basically just lumpy, so just placed on the dish. And extrapolating from this analogy, if we are to use materials which undergo phase separation or dynamic aggregation to form fibers, we then need to think about whether we need to strike a compromise between fiber organization and fiber functionality. So again, sometimes we need to make this compromise dependent on the application. So with this in mind, the goal of our fiber printing technology development is largely focused on two main areas. First is to really optimize the fiber material property. This means that to adjust the fiber spinning condition, the design of the nozzle, to print materials at a much reduced voltages or no voltages, or at a mild temperature, so that we can enhance the material's properties. By enhancing the material's properties and creating functional fibers, we then need to think about connecting these fiber materials to a device. And therefore, the second goal is the device connection. So with this, we can potentially create fiber integrated cell culture and also inbuilt fiber sensors. So how we get started? So back in 2016, we developed a process called the low voltage electrospinning, or LEP. So in the process of LEP, we decrease the operation voltage of solution electrospinning from kilovolts to below 100 volts. And in more recent research, we combine the LEP process, the low voltage electrospinning process, with 3D plastic printing. So we can start to design fiber patterning in a CAD, a CAD file, like conventional 3D printing. So in this video, it shows that the blue are thermoplastic printing, and also the green, they represent the fiber printing. So we can start to create architectures in a layer by layer fashions using fibers. So in all, this becomes an additive batch spinning process, a new process flow of 3D printing. Using the additive electrospinning process, we could design cell culture devices with fiber patterns and use them to guide the growth of cellular aggregates. Because fibers are printed in suspended fashion, so as you can see here, very large spheroids can be created and they can be guided to form on the fiber structures. So very interesting is that using the property of the hydrogel fibers, so if we are to print hydrogels on these devices, and because the hydrogel two ends are being constrained, these fibers can swell in water and become relaxed. So growing the cell aggregation on the fiber, the cells will start being able to buckle the fiber and crumble the fibers. And a theoretician come to see these images and say, oh, that's very interesting, because the conformation of the fiber structure, so this curved fiber structure evolves as the cells aggregate and also exert forces on the fiber. And therefore, by knowing the mechanical property of the hydrogel fibers and looking at the conformation of this buckling, one can actually estimate the forces, the traction force exerted by the cancer cells on the fiber. So effectively, the soft hydrogel fibers are acting as a force probe. The theory proposed is proposed in this paper, which you can have a look if you're interested. So the benefit of using solution electrospinning is that by choosing the right solvent, you can process a large range of functional materials. Here in these two papers, we show that we can load very high concentration of decellularized matrices into membrane scaffolds. And these membrane scaffolds can be patterned into tunable mesh size. So in some cases, for example, in the case that I'm showing you here, this is a very thin semi-transparent membrane, very fragile, made of gelatin composite with decellularized matrices. And if we were to print this membrane on a substrate and lift it off, and it cannot be lifted or transferred. So the process here is that we actually directly create the membrane on the 3D printed ring and then make it into a device for cell culturing. So here on the right-hand side, you can see that we demonstrated a co-culture model between glomerulus endothelial cells and also podocytes in order to mimic the glomerulus barriers, the trilayer glomerulus barriers seen in the kidney structure. So the next fiber production technique recently developed in my lab is called the dynamic near field electrospinning, or the DNFES. In the DNFES process, the distribution of static field induces a dynamic switching between the linear jetting and also the controlled whipping behavior. So as shown in this video here, so you can see the linear jetting and also the control whipping process. During the control whipping process, it actually pulls the piezoelectric alignment director as well as forming a fiber that bridging the two electrodes. So we use this nanofiber to create a spider-inspired, non-resonant acoustic sensor. Therefore, we can harness some pulses to fiber vibration and then to electrical energy. The sensor has a very high sensitivity and energy conversion efficiency from majority of the audible sound spectrum, as shown by this characterization graph here. More importantly, this performance is achieved at a hearing-safe loudness level, which is a big advantage compared to established PVDF fiber structure, which will require a much higher level of acoustic pressure in order to achieve a reasonable level of signals. Now, working with Clemens Cummins' group from Cambridge, we use the fiber printing process to guide the self-assembly and patterning of amyloid fibers. This solution process technique is very mild, and therefore it can retain the intrinsic fluorescence of the amyloid beta sheet folding. The mechanical properties of the fibers can be tuned at a single fiber level. As you can see here, in the blended system, the fibers are very brittle. While in the co-shaft system, the fiber is elastic. With this, we can start to bring the nanoscale self-assembled fibers, which have been very difficult to manipulate, to up to centimeter-long fibers with bulk performance. Here, parallel fibers, very fine, they like a little bit like spider silk in their appearances, are patterned between a gap structure, and then this rice grain in comparison being loaded onto the fibers, and you can see the highly ductile and elastic structure of the fibers. So recently, we pushed the technology further to create core-shell fiber structures in a process called in-flight fiber printing, or we call it IFP. IFP combines fiber synthesis, patterning, and bonding in a single step. We use this technique to print silver fibers through in-situ reaction without post-processing. And the silver fibers have very high performance. They are very conductive, we have small diameter, and therefore we demonstrated their ability to suspend LED structures, small LED structures. Because these fibers don't have any substrate, and so they look even more transparent than the transparent conducting film, such as ITO, which is backed by like a film. And they also don't have any reflections. And subsequently, we also demonstrate the possibility to form a 3D optoelectronic device, such as the floating LED structure that you see here. So another capability of the IFP is to interconnect fibers of vastly different properties, from organic to inorganic. So inorganic fibers represented by the silver fibers here, and organic fibers represented by the P.PSS fiber can be merged together, form either homojunction or heterojunction structures. This is by exploiting the use of a very small voltage to pull the second layer semi-solidified fiber to the first layer structure. And then if we don't have a voltage, the two junctions won't be merged. And if we apply small voltage, the two fibers will merge into junction structures. So of course, then we will build these fibers into a device structures to create sensing devices. So one interesting fact about P.PSS is that the resistance of P.PSS fibers is moisture sensitive. Here, Andy, the first author from the paper, is wearing one of the printed fiber sensors on his mask. And the aim is to detect the moisture, the breath leakages from his mask, because the human's breath contains about 90% moisture level. And so here is the ring structure, and the fiber is basically in between this ring, which you can't really see with your naked eye. And here is the SEM image of the fiber structure. So due to the small diameter and suspended nature of the fibers, the device shows at least 6 times better response than a commercial sensor in distinguishing individual breath leakages. So what's interesting is if you look at this graph here, when a repeated fast breath is applied on the sensor, simulating the condition that the person is breathless due to respiratory disease, the commercial sensor cannot distinguish any of the individual breaths, but our sensor can pick up all the signals. So by stacking the fibers in layers and by making each layer of fiber with a different material, a multimodal and spatial sensing performance can be designed. For example, by stacking piezoelectric fibers with P.PSS fibers, both sound and respiration information can be acquired from the same sensor. And so here is the tri-layer sensor that's produced with different functions across each layer, and it's really small, that can be attached to the phone. And we use this to measure the different coughing and also exhale conditions. And in a different application, and probably more relevant to the biofabrication community here, P.PSS fibers, when they made into a high resolution, they can be used to guide the attachment and alignment of cells. So the fibers remain stable and functional in culture media for more than 2 weeks. And they could be used to form an impeded metric sensor to monitor cell dynamics. Here in this video, we can correlate the cell behavior from the attachment to the alignment with respect to the impedance measurement in real time. And this sort of devices could start to open up new capability for bioelectronic sensing using biocompatible fibers. And so now, taking a break from the technology development, I would like to move on to the next topic, which is to look at a case of cell fiber pattern interaction for gaining physical insights. Cell migration plays an important role in physiological and pathological processes where the fibrous morphology of the extracellular matrices could regulate the migration dynamics. To mimic the morphological characteristics of fibrous matrices, straight, wavy, and grid fiber patterns were fabricated with polystyrene. And polystyrene is basically the same material constitute of a Petri dish. Here are some typical videos showing the migration process on these fiber patterns. And the cells we used here is MB231, a highly migratory breast cancer cell line. So in the first case, the cells would go along the loops or the curves of the fibers. In the second case, the cell's movement seems to be restricted by the boxes. And in the last case, the cells were initially guided by the along fiber, but then its migration direction was diverted when it missed an orthogonal fiber. Yeah, so let's wait until it, yes, there you go. When it missed the orthogonal fiber, its direction is changed. So what we are interested to understand here is whether there are any guiding principles where cell migration could be affected by the fiber pattern contours. As you've seen in the video, clearly the cells can dynamically adjust their shapes in response to different fiber environment during migration. Therefore, we adopt the concept of aspect ratio, which is used as an indicator for cell shape. If we were to fit the cells with an ellipse, the aspect ratio is defined by the minor axis length divided by the major axis length of this ellipse. So as the fiber pattern changes from linear to loop, the cell's aspect ratio increases and becomes more circular. However, when we compare the average step speed for the single cell migration at different fiber patterns, the step speed is actually quite unchanged with respect to the different fiber patterns. So we suggest an explanation for this is like a person, this green person here walking on a track. So the global contour of the track determines the direction of the walker, but the step speed of the walker is dependent on the person's ability and also the friction between its foot and the road. But this is more like the intrinsic ability rather than the global contour of the track. Therefore, we propose that the global fiber pattern determines the cell migration direction and cell shape, but not the migration step speed. An even more interesting finding was that for all the patterns we looked at, the cancer cells were prevented from path diversion when the pattern feature size is greater than the upper limit of the cell's minor axis length. So here in the red defines the pattern feature size, and the upper limit of the minor axis is how far this minor axis can be stretched up to. So I've tried to find an analogy for this and bumped into this koala image. So this is provided by the Queensland Tourism website. So let's say the koala is currently at the upper limit of its minor axis length, shown in red. Based on the koala's shape and ability, its body is able to move between the branching when the branching spacing is spacing 1. However, the koala will find it difficult to move between the two branches at the space number 2. So coming back to the microscopic scale, our finding would suggest that although cells can dynamically adjust their shape in response to different fiber environment, their ability to divert from the existing fiber track is limited by the cell's ability to stretch away from their existing fiber. And this ability is reflected by the upper limit of the cell's minor axis length. And in the case of MB231, the upper limit of the cell's minor axis length is 30 to 35 micron. And this governs the feature sizes of the fibers beyond which the cells would not be able to divert their paths. And this is consistent for all the different conformation of global fiber patterns that we studied. Now, moving on to the third part of my talk, I would like to introduce you to a new organoid engineering technique called MOrPH. Since some of the processes discovered are quite unexpected, this leads to my question here, when we should engineering our system and when we should just let the biology to do its own thing. So the MOrPH process stands for multi-organoid patterning and fusion. In our recent publication, we use the MOrPH process to assemble scaffold-free life-size organoid tubes for organoid on the chip and also branching tubular structures. This work is performed in conjunction with my colleague Zhou Hinli in the Cambridge Stem Cell Institute. So organoids are miniature collection of cells which can mimic the biological features of an organ. Airway organoids can be developed from single basal stem cells when growing metrogel, but they grow in different sizes and are relatively small. In the MOrPH process, we release individual airway organoids from the metrogel and put them into an aggregate. So here, and we put them into the aggregate, and then put them into a microwell so that their shape can be patterned. Next, this cluster is released from the metrogel. Sorry, this cluster is released from the microwell to grow in a floating culture. And over the next 2 weeks, the organoid aggregates would then undergo inter-organoid surface integration, as shown by this video. So you can just about to see a protrusion is now extended from an existing multi-organoid cluster and grab another small organoid onto forming an integrated surface. Now imagine a cluster of soap bubbles which can stick together. The big challenge of this cluster to transform in a single gigantic bubble is to get rid of the partitioning walls. So how can we do that using engineering? So we discovered that as the organoid aggregates was growing in a floating culture and under the right condition, the unwanted cells forming the partition can naturally be extruded out. So this is very interesting because we have not done any engineering, but naturally the cells will be able to get rid of the unwanted cells forming the partition. And through this luminoclearance process, a connecting lumen structure can be established without any engineering imposed. So the shape of the microwell defines the shape of the organoid tube. With the process of patterning and fusion, the final fused product will have a defined tubular shape and a single lumen structure. Some of these tubes, the one shown here with the aspiration of 3, it has a size which is about 300 times compared to the average size of an individual organoid. We also assess the cellular morphology and epithelial composition of the organoid aggregates at different stages. Taken together, our data suggests that upon metrogel depletion and organoid fusion in the floating culture, the fused tube retains a single epithelial layer of basal cell identity without pseudo-stratified epithelial morphology. They also show changes in epithelial polarity. So in the future work, it is important to perform systematical study to find out a more balanced matrix concentration to further optimize the biological property of the fused organoids. And so the matrix, again, is very important, forming part of the equation on how to develop a more biological functional macro-scale organ structures. So our work on organoid fusion also made me to look back at the key points when engineering actually has been taken place. Engineering was applied at the beginning, the aggregation stage and aggregation and patterning stage, which gave the initial boundary conditions for the cells to develop into. But for the rest, and majority of the time, for the rest about 3 weeks, the cells actually figure out what to do by themselves based on these preliminary conditions defined. So the ability to make big organoid tubular structure also open up the possibility for further downstream applications. This includes scalable organoid device integration, multi-tissue integration, and also construction of hierarchical fusion. So as an example, by generating an organoid tube of the size similar to a mouse trachea, we can connect this tube to a perfusion system. You can see the tube exhibited cyclic lumen expansion and relaxation with respect to the input flow. Hierarchical fusion can also be adapted to form branching tubular structure, which is actually very difficult to establish, especially in an organoid system, as shown by the right-hand side. This biological assembly approach will be very interesting for fundamental biological studies and further could act as a new drug screening tools for respiratory disease. Now, in the final topic, I would like to talk about 3D printing biomimetic cochlear and its integration with machine learning for clinical informatics. This work was performed in conjunction with my clinical collaborator, Manhill Banks, who is a professor of otology and scalp-based surgery at Cambridge. And Ip Man Lei, the first author of this paper, presented some of this material at the recent ISBF Twitter conference. Another reason to join the community. In this talk, I will expand a little bit on my personal perspective as well. So cochlear implants are by far the most widely used neuromodulation electronic implants. And it is this tiny device here that inserted into the inner ear, the cochlea of the patient. So the cochlear implant is designed to restore a patient's sound perception by injecting electrical current at defined location along the cochlea. A major limitation of today's cochlear implant is their imprecise control of stimulation due to the intrinsic conductive nature of the biological tissues. And my clinical collaborator will call this problem the current spread problem. So understand current spread and how it could correlate patient-dependent factors is hampered by the poor accessibility of the inner ear. And therefore, having a suitable model system, which we hypothesize to simulate the electroanatomy features of the human cochlea, is very important to address this question. Now, overlooking the different established model systems, from animal models to finite element analysis to cadaver models, all have their limitations in replicating the electroanatomical characteristics of living human cochlea. So our work, the aim is to use 3D printing and machine learning to model the relationship between the anatomical features of the cochlea and also EFI, and so that we can predict patients' clinical readouts. So what's EFI? EFI is a clinical readout that shows the voltage distribution along the cochlear implant array inside the patient's cochlea. Here is an EFI spectrum that's shown on the right-hand side. So this is related to a very important question about our study design. What kind of patient data can we use? So this flowchart here shows the standard of care clinical procedure. It starts by acquiring a patient's CT scan of the cochlea. And then based on the information on the CT scan, the surgeon will insert a cochlear implant. And after the implantation, an intraoperative EFI would be acquired on the patient. Now, for producing a model to evaluate how electroanatomy of the patient cochlea affects EFI, there are two major challenges. First, the cochlea is a very small feature. And as you can see in this preclinical CT scan, that the patient's CT scan can only resolve the ensemble cavity of the cochlea, but no any fine details or the partitions can be resolved in the CT scan. Second, we have no idea about the electrical property of the cochlear tissue in a living person. In fact, how can you measure the electrical property of the cochlea in a living person without actually using an intrusive process? So to tackle the first challenge, instead of following the conventional procedure of copying the CT scan reconstruction in the 3D printing, we use 4 geometric parameters to describe the cochlea. So this includes the basal lumen diameter, taper ratio, width and height of the cochlea. And to address the second question, the cochlear tissue resistivity, we choose replicate the human scale resistivity, which has been measured in living human. So because the human scale is actually on the exterior, so it's easy to gain the in vivo measurement. But we choose not just to replicate a single value of resistivity. We choose to replicate as wide range possible the reported values of resistivity in the literature, so that we can make sure potentially our cochlear tissue resistivity will fall within this range. So here is the video showing the printing process, where we take the inspiration from the porous bone structure to construct the electromimetic bone matrix. So this consists of a PDMS forming the insulating phase and also channels forming the conductive phase. And subsequently, we print a cochlear duct inside this matrix structure, following the 4 geometric parameters that defined previously. So here it shows the finished product. This is the interconnected porosity in the matrix, where we then would be filled it with physiological sealing and the cochlear duct lumen. And finally, we have inserted a cochlear implant array into the cochlear structure. Now, by varying the void density in the PDMS, we can match the wide range of resistivity values of the human scale reported. So before we settle on PDMS, actually we tried a range of hydrogel materials as well as hydrogel with filler matrices, but none of them was able to reproduce the full range of the resistivity values that we are looking for the in vivo value of scale. Now, an added benefit of using a porous PDMS matrix is that it has adequate mechanical property, which is stiff enough for a guided implant insertion, but it is actually 4 orders of magnitude softer than bone. This really matters, because if the matrix is actually as hard as bone, the electrode, the cochlear implant electrode, would have been damaged just after a couple of mock implantation experiments. And I was told by my clinical collaborator that each cochlear implant has a market price of £10,000 or above. So we'd better make a model that don't damage the electrodes during experiments. So using the impedance spectroscopy, we can also show that an electromimetic bone matrix can be designed to have a similar frequency-dependent electrical property as the cochlear bone measured in a cadaver human head. And with this foundation, my student Ik then printed a library of biomimetic cochlear, which could capture the diverse geometries of human cochlear lumen and with a range of resistivity reported by, for the human scale. Then a cochlear implant electrode is inserted into the model to measure EFI to simulate a clinical scenario. Finally, a neural network model is established by the training data acquired in the 3D printed cochlear models. And so this entire process, we call it 3D printing neural network co-modeling, or 3PNN. So without going into details, here are the selected results and performance of 3PNN. First, the 3D printing model can reproduce a range of clinical observed EFI, including the mean EFI of 97 patients where we have no CT scan available due to ethic restrictions. And second, for those where we can obtain the ethical approval to correlate the patient CT scan and EFI data, 3PNN provides prediction for 28 out of 31 patients. A further application is demonstrated by using inverse prediction capability of 3PNN for on-demand 3D printing. Again, due to ethic restriction, it is often not possible to have paired patient CT and EFI data. However, simply based on patient's EFI, we can do some back envelope calculation and predict the most likely the patient cochlear geometry. And then we can print this cochlear geometry and reproduce the EFI. So as shown in here, the EFI measure in the 3D printing model compares very well with the patient's native data. So using 3PNN, we can also gain new insights and discoveries. First, our results suggested that GFR is largely controlled by volumetric conduction of the cochlea, and this confirms our starting hypothesis that the dominating factor which determines the current spread is the electroanatomy of the cochlea. We can also now go back to our motivation. What are the patient-dependent factors causing current spread? Our findings shows that the cochlea with larger basal lumen diameter and also less tapered lumen will have more pronounced current spread. Finally, we can deduce the in vivo cochlear tissue resistivity. So this is about 6.6 kilo ohm centimeter. And to our knowledge, this is for the first time such a value was reported. So to summarize this session, the 3PNN provides a print and learn modeling concept where a physical model can be used, which is ethical and economical. The approach that we took in our study was that we start with a set of our hypotheses, and we would like to use this hypothesis to test the clinical data where we can obtain and compare to. And this is very important because in the lab, you can have a high resolution imaging, you can have many tests that you can do. But later on, the models may not be correlated or validated against patient data because of the test mismatching. And being able to consider clinical data from the start will also help to translate the research finding into the clinics. Finally, we are all aware of the tremendous power of artificial intelligence, or AI. But AI needs to start its learning from a good set of data. Because of patient privacy, patient safety, and ethical concerns, well-characterized clinical data is hard to combine. Therefore, 3D printing may be a powerful tool to create physical models, which might create well-characterized training data with purpose-built surrogate to clinical data for machine learning. So, yes, with this, I would like to conclude my talk to have a look at an overview of where we are now in terms of biofabrication development. So I would like to come back to this aspiration set up at the beginning of my talk. For the phase of developing organoids and engineering living systems, my lab has shown the MOrPH process for guiding the upsizing and shaping of airway organoid tubes. This approach might open up the possibility to make tubular structures that integrate biology, anatomy, and physiology in the same tissue engineering product. For the in vitro modeling and disease modeling area, our lab has been working on a microvessel on a chip model and a brain cancer on a chip model. Our aims were to optimize the relevant biological culture conditions, and I did not have time to talk about them today, and here are a couple of references which you can refer to. In the area of scaffold building, we focus on creating fiber structures with different materials to enrich functionalities. The potential is to intertwine functional fiber structures with biology to create hybrid systems. And lastly, in the area of non-biological model development, we demonstrate the power of 3D printing and machine learning co-modeling. This allows the 3D printing model system to go beyond surgical modeling and implant testing, but actually to discover new knowledge for patient and for the human body. So with this, I would like to thank my group members and my collaborators who contributed to the results and fundings that I talked to you today. Due to the global pandemic, some of my group members have not actually been home or see their family in person for 2 years. And so it's a tremendous commitment and a dedication for them to work in my lab in Cambridge. So I would also like to thank the people and the funding agents who provided grant support to my work. And of course, I would like to thank the International Society for Biofabrication, who provides the platform for us to come together to listen to this conference. And so, yes, and please join the society, and here's the information which you need to know. And thank you. So questions. I'm happy to answer any questions that you may have.
Thank you very much, Shery. Thanks for the very nice and extensive talk on your brilliant work. We have a few questions already. We can start with the ones in the chat, maybe. So the first question, I'll go in order for how they've been posted. The first one is from Gianluca Cidonio. And he would like to know, how are you able to move around and precisely position the organoids for the formation of the MOA?
Yes, so we didn't need to. We literally just put them in a microwell.
Okay.
So yes, I understand the. So we also work on this pick and place method in our lab on a separate project. But for organoid, we find that we don't need to engineer them too much. And actually, it's not good to engineer them too much because they start to feel a bit under the weather. Yeah.
Okay, thank you. And the second question is from Shuzi Suzanne. And she has actually two questions. So one is, how do you relate the cell migration dynamics along the polyethylene fibers to what happens in vivo, the biological migration in vivo? And the follow-up question is still on the in vitro and vivo relationship, sort of. In this case, on how you relate the biological function of the organoids formed by aggregation in your structures to the in vivo function.
Yes. So the fiber structure migration actually is for the in vitro model, you will have to model one aspect. And cell migration in vivo not only has got global topology guidance, but many other factors. Okay, so but I believe that there's a simpler question to answer this is whether this CORSI 2D system can be converted into a 3D when your fiber networks are actually packed together quite randomly. I think the same thing will work. Simply because imagine a person like the koala system that I kind of show you is a 3D, it's kind of 3D structure. And what's interesting is how you structure your body. It depends on how you define your minor acids, but it has to be to do with the maximum capability of that object to be stretched away from your existing attachment. I think this will work in 3D as well. Yeah, but for in general, in terms of migration, as you know probably as well, is there are many other factors in addition to the global topology that controls the migration. So I think the second question is how much is the resolution range of this technique for the fiber spinning? Right. So depends on the material, but it can be 100 nanometer to 10 micron on above. So the second question is the second question is the bending instability. Is that the question?
Yeah. The second question was the video of the fiber spinning. The gel still presented bending instability. So how could the fiber deposition were well controlled on the substrate? That was the second question by Feng Lei.
Okay. So the for the bending instability, you mean which process? Which is the LEP process or the dynamic near field electrospinning process? Okay, so I will try to answer it. So there are two different processes here. In the LEP process, there's no bending instability. So in the low voltage electrospinning process, it's a linear jet, so we operate in the linear regime. In the other process where I turn dynamic near field electrospinning, where I use it to pattern the piezo electric fibers, there is bending instability on the plane where the fiber switch between the two electrodes, and we need that bending instability, a control bending instability, so that it can attach to two electrodes, because the pattern is actually orthogonal. The plane of pattern is orthogonal to the stable jet. I hope I've answered your question. So
actually, the second question by Susan's one, there was the relationship between the biological function of the organoids and formed by aggregates to the in vivo function.
Yes. So so as I mentioned, and you can actually look at our paper, is that the in vivo function, because we use a mouse organoid, is quite well characterized. And so what we found that is when we undergo the fusion is that all the basal stem cell, so in the mouse trachea, there's basal stem cell, and on top of the basal stem cell, there are some differentiated cells. And so the basal stem cells are all there. But then the top layer, the differentiated cells, reduces during the fusion process. And so, but we also, as in the paper, which we didn't talk about today, is that we compare it to Metrogel encapsulated version, which reduces the fusion efficiency, but in that version, that's able to maintain this polarity and maintain the differentiated cells on top of the basal stem cell. And so what I'm guessing is really depends on the application, because if the aim is actually to just use the basal stem cell, which can differentiate further in, for example, in vivo during implantation, there's no need for, well, it depends, but yes, but then there might be not as requirement to have this very well organized structure like in vivo. But for drug and toxicity testing, of course, you will need a very well organized structure, and therefore the extracellular matrices will be required. And so we're still working on optimizing the protocol so that we can have a balanced fusion efficiency and also the biological characteristics.
All right, thank you for your answers. Andy may have a question on your presentation, right?
Yeah, thanks, It was really nice work, Shery, particularly on the organoid fusion. I was kind of curious, I imagine that it's very scalable, like in the longitudinal direction of a tube. I was wondering how many, I guess in the radial direction, how many do you think you're fusing together? And can like the cells in the center, say, of after you jam them together, can they kind of, I guess, migrate into that tube on the outside? I hope if that makes sense. Hopefully.
Yes. Yes. So so in the paper, we also kind of examine this process. The organoids, they are actually fluid inside. So the organoid we work with, if the airway organoids, they have it's like a bubbles, but then you imagine like a balloon filled with water. And so this is the organoid structure that we work with. And therefore, the cells are actually a little bit like floating inside. And so the shell is actually being able to, what we found that under the right condition, the cell can open, the shell of the organoid can open and release this, the cells that don't want. They extrude it out of the shell naturally. And so, but then I guess in other research, it also shown that naturally the organoids, if it has got fluid center, due to the continuous pumping of the fluid, some weak points on the outside will be able to open, and then the inner cells, unwanted cells, will be able to release. So it's a balance between fluid pressure and also the opening of the epithelial external layer. So.
Okay. That makes sense. Do we have? I don't think there's any more in the chat. Does any of the other panelists have any questions?
There is one more question in the chat.
Oh, sorry.
Yes. So I think Diane has asked whether I can explain MORPH and MOA structure in more detail. I wonder how the shear stress of fluid movement can prevent adhesion between surface cell layer, even though the size of the organoid and volume of the lumen are large. Yes. So I'm trying to guess.
Dayon, do you mean that even though the organoids are large, they can still move quite a lot because of the shaking in the fluid, and then that will make it difficult the fusion. Is that that's what I understand, but I'm not sure. Can you confirm, Dayun? Or maybe we can, maybe you can give your interpretation, Shery.
The question is what Ricardo you suggested, and then if you see we place this organoid in like a well, but then we also add another gel. So I hope that answers your question, and so that kind of like put the tubular structure down.
So there is not much chance for the organoids to move around. They're actually partially confined.
Yes, so they are growing in floating culture, but when we need to use them, we then put them onto in a gel structure. Yes.
Okay, thanks.
Yeah, no problem.
Yeah, thank you very much, Shery. I think we should wrap up here just because we just up time. So thank you very much for a very brilliant presentation. A lot of information to take. Thank you very much for everyone who has joined us today. We are having our next webinar in 2 weeks, so it will be on the 5th of August. We will announce it on Twitter and on social media. And our next speaker will be Professor Sang-Jin Lee from the Wake Forest Institute of Regenerative Medicine in US. So please join us for our next meeting, and also join us to thank Shery for picking up our invitation and presenting this really great presentation. Thank you very much.
Yeah, okay, thank you. Yeah, drop me an email if you have any more questions, so that's okay. Yeah, thank you. Bye now.
Bye,
Thank you.
everyone.